Welcome to the third lecture of week 7. Today I am going to discuss about citric acid cycle and oxidative phosphorylation. So, we have already seen this in the previous lecture this week, that glucose is metabolized to pyruvate, right? And in this process, oxidation of glucose, we got 2 ATP molecule synthesis, right? Now, glucose can be oxidized all the way to carbon dioxide and water, but in this case there is only one oxidation step that happens here in step 6, and we get some amount of energy extracted out from glucose and we have it stored in ATP and this high energy molecule which is pyruvate. But the ultimate goal of oxidation is to convert glucose into carbon dioxide. The electrons that come out, because this is oxidation, so glucose will give up electrons. Those electrons will be accepted by oxygen to produce water, and we will see that this happens as 2 different sets of half reactions. So, in one set glucose will get converted to carbon dioxide, in another set oxygen will accept all the electrons and will form water, and this happens in more subsequent steps, and finally all the energy are stored in the formation of ATP, right. So, this slow oxidation process that happens inside a cell are called cellular respiration. So, we generally call it respiration, but since this process is happening inside the cell, we call it cellular respiration, and we will see that it will have 2 further stages. One is citric acid cycle and the next one is oxidative phosphorylation. And it is important to point out that the subsequent steps that happens after pyruvate synthesis are much more complex. The reactions are more complex, and of course they need an oxidation environment. So, it is expected or it is hypothesized that the later part of these reactions evolved once cyanobacteria were evolved, because they started producing oxygen and then the environment became oxidizing, and then only these more oxidative reactions evolved. Okay. So, this is the complete summary of what we are going to study today. So, we have already seen glucose to pyruvate formation. So, that is glycolysis. There were 10 reaction steps. Only one oxidation step was there where the electron was accepted by NAD plus to form NADH, right. Next, we will see that pyruvate gets converted to acetyl coenzyme A. So, this another oxidation step. So, another electron pair will be accepted and by NAD plus to form NADH, and one molecule of carbon dioxide will go out. So, now we will also keep track of the carbons in the glucose. So, glucose was 6 carbon molecule. From one glucose 2 pyruvic acids were formed. So, pyruvic acid is 3 carbon molecule, right. Now, if we consider pyruvate, which is 3 carbon, in this step 1 carbon goes out. So, we are left with 2 carbons, which is the acetyl group, right. Then this acetyl group as a thioester, acetyl CoA, goes into the citric acid cycle, and here we will see that further oxidation steps happen where 2 more carbon dioxides go out. So, acetyl, there were 2 carbons and they are now released as carbon dioxide. So, pyruvic acid gets completely oxidized as carbon dioxide 1 molecule, 2 molecule, 3 molecules. So, all 3 carbons are released, right. And the energy is mostly stored in different forms, majorly in the form of NADH and FADH2, right. So, these are the electron acceptors, right. So, now the remaining half reaction is still there, which is oxygen will have to accept these electrons which are in this reduced species to form water. So, oxygen has to form water, and that happens in stage 3, which is called oxidative phosphorylation, right. And we will see that most of these reactions, so starting from pyruvate to this, all of these reactions occur mostly inside mitochondria. So, glycolysis, so glucose to pyruvate, this happened inside cytosol, and from here to here most of these reactions will occur inside mitochondria. So, I will talk about mitochondria in more details in the later part of this lecture. So, now I am going to focus on this second. So, we have already looked at stage 1, which is glucose to pyruvate. Now, we are going to look at this later part of stage 1, which is oxidation of pyruvate to form acetyl coenzyme A. the enzyme that is involved in this step is called pyruvate dehydrogenase. Okay. So, it is not just one enzyme, there are 3 enzymes E1, E2 and E3, that is why it is called a pyruvate dehydrogenase complex. Right. Okay. So, this is the reaction. This is pyruvic acid. This acidic group goes out as carbon dioxide. So, we are left with this acetyl group CH3CO, which is taken up by this coenzyme, acetyl coenzyme. This is the coenzyme A. So, when it takes up the acetyl group, it becomes acetyl coenzyme A. So, that is the coenzyme A and this is the acetyl group, right. Now, pyruvate dehydrogenase is the enzyme complex which catalyzes this reaction, and it consists of 3 different enzymes termed as E1, E2 and E3. And these 3 enzymes, they use all these different cofactors. So, there is acetyl coenzyme A, there is coenzyme A which becomes acetyl coenzyme A. There is NAD plus which becomes NADH, so it accepts the electrons. There is TPP, there is lipoate and there is FAD which will become FADH2 and then it will get reduced to FAD. So, 1, 2, 3, 4, 5 different cofactors are used by these 3 enzymes in this enzyme complex. So, here is the structure of coenzyme A. So, this is coenzyme A. So, you can see that there is adenine, there is ribose 3-phosphate, there is the phosphate group. This is called pantothenic acid, and finally, there is this beta markapto ethaneamine group, right. So, all of these we will write as CoA, coenzyme A, right, because the reaction center or the active site is this thiol group. This is the reactive thiol group. So, we will write all of this as CoASH, right, and when this thiol reacts with the pyruvate to form this is the acetyl coenzyme A. This is the lipoic acid, the other cofactor that we have not seen before. So, you can see that there are all these different parts. So, this is connected to the side chain. So, this is E2 enzyme. So, there are 3 enzymes E1, E2 and E3. This is a lysine of the enzyme E2, right. So, this is the lysine side chain which will be NH3 plus, but it reacts with this carboxylic group. So, it forms sort of an amide bond, right, and there is a disulfide linkage here, right. So, this disulfide is very similar to the disulfide that we have seen for cysteine amino acid side chains, right. So, this is the oxidized form and this can get reduced to form SH and SH, right. So, this is very similar to the disulfide oxidized and reduced form of cysteines, right. Now, this free SH can take up the acetyl group from the acetyl coenzyme A to form this acetylated form. So, we will see all these 3 in the reaction that is catalyzed by this pyruvate dehydrogenase complex. The enzyme complex looks something like this. Okay. So, E1 is one enzyme, E2 is the other enzyme and E3 is the third enzyme. So, there are multiple copies of these enzymes and they together form this complex structure, right. Now, when reactions happen, E1 takes up the substrate, it is passed on to E2 and then finally it is passed on to E3. So, in any of these steps, the substrates or the intermediates, they never diffuse out. So, they are always attached to this enzyme complex, right, and that is something that we will refer to as substrate tunneling. So, let us see what happens. This is pyruvate, right. So, pyruvate comes in and it interacts with E1. So, this is E1 which is yellow, this part. The middle part is E2 which is this green part, and the last one is E3 which is this red part, right. So, E1, E2 and E3. So, E1 catalyzes the first step where TPP, thiamine pyrophosphate, interacts with pyruvic acid, carbon dioxide is released and this acetyl group is attached to TPP as hydroxyethyl TPP, right. Now, TPP is regenerated and this hydroxyethyl TPP is oxidized to acetyl group, right. And when that oxidation happens, so this lipolysine, this is present in the oxidized form, so it gets reduced. So, this gets reduced and this gets oxidized, right. So, we have this acetyl lipolysine. Now, this arm, it is a very flexible part as we have seen in the structure. So, you can see it in this structure. So, this is a long chain. So, it is a very flexible part. So, it swings from this position to this position, right. And when it does that, this acetyl group is handed over to coenzyme A, right. So, now coenzyme A becomes acetyl coenzyme A, and we have this reduced lipolysine. Now, this reduced lipolysine has to be regenerated into the oxidized form. That is done by FAD. So, FAD gets reduced to FADH2, and this reduced lipolysine gets oxidized to oxidized lipolysine, right. So, now it is again ready for the next round of the reactions. FADOH2 is the one which has ultimately accepted the electrons that have been released by pyruvate. Now, it will hand over the electrons to NAD plus, and it will itself get regenerated. So, this is the E3 complex, and NAD plus gets reduced to NADH. So, this is done by the third enzyme, right. So, all of this TPP is regenerated, oxidized lipolysine is regenerated, FAD gets converted to FADH2, and then it goes back to FAD. So, it is regenerated. NAD plus gets reduced to NADH, and coenzyme A accepts the acetyl group. So, it becomes acetyl coenzyme A. So, these are the two end products of this reaction. So, we get acetyl coenzyme A, we get carbon dioxide, and we get NADH. Release of carbon dioxide drives this reaction forward. So, this is the summary of the acetyl coenzyme A synthesis by pyruvate dehydrogenase. Pyruvate dehydrogenase complex, in short we will refer to it as PDH. It is composed of multiple copies of 3 enzymes. Okay. So, these 3 enzymes are pyruvate dehydrogenase, which is E1, dihydrolipoyl transacetylase, which is E2, and dihydrolipoyl dehydrogenase, which is E3. Now, if you see the structure, you can see that it is obvious that there are multiple copies of these 3 enzymes are present in the PDH complex. There are 5 different coenzymes that are used by PDH: thiamine pyrophosphate, which is TPP, flavin adenine dinucleotide, which is FAD, coenzyme A, nicotinamide adenine dinucleotide or NAD, and lipoate. So, this is a new coenzyme that we have seen here. E1 catalyzes the first decarboxylation of pyruvate. So, this is the first time a carbon dioxide is produced. We will see 2 more carbon dioxide production in the citric acid cycle. The acetyl group is transferred to one of the SH groups of the reduced lipoate. E2 catalyzes transfer of this acetyl group to the coenzyme A. So, from the reduced lipoate, it is transferred to the coenzyme A, which results in the formation of acetyl coenzyme A, right. E3 regenerates the oxidized lipoate, right. So, it regenerates the disulfide oxidized form of the lipoate, and the electrons are transferred to FAD and finally to NAD+. So, NADH is formed. The long lipolysine arm swings from the active site of E1 to E2 to E3, right, and that is something that is critical for this catalytic cycle, and it keeps tethering the intermediate to the enzyme complex, which is something we refer to as substrate channeling, right. The substrate never diffuses out, right. So, it is always bound to the enzyme complex, and this is what we call substrate channeling. Glycolysis occurs in cytoplasm. In eukaryotes, the acetyl CoA formation and subsequent reactions take place in mitochondria. So, this is something that I will keep coming back to again and again. Okay. So, now citric acid cycle, right. So, this is where we are. Acetyl coenzyme A has formed. Now, this acetyl coenzyme A will go into this cyclic reactions, right, which is referred to as the citric acid cycle, because the first product that is formed is citric acid, and these reactions go in a cyclic fashion. So, there are 8 steps. We will go through these 8 steps, and we will see that it goes on in this cycle. Now, another important feature of this cycle is that many of these products that are formed are taken up by different metabolic pathways. So, I will show you one slide where all those pathways are highlighted. So, let us look at the citric acid cycle. The first step, acetyl coenzyme A, it reacts with the last step, right. So, the product of the last step, which is oxaloacetate. So we have acetyl coenzyme A here, we have oxaloacetate here. This condensation reaction is catalyzed by citrate synthase, okay, resulting in the formation of citrate. So this is citric acid, and the coenzyme A is regenerated. So this can again go back to the previous step to form more acetyl coenzyme A. So it is recycled. After the first step, we have citrate. Citrate will be converted to isocitrate by the enzyme achotinase. Now achotinase, it converts citrate to isocitrate via an intermediate which is called cis-achotinate, right. So you see that a water molecule is taken up, so water molecule goes out here, so this OH and H from here goes out, so it forms this double bond. Now water molecule is added, but now OH is added here and H is added here, so this becomes isocitrate, right. So this is the second enzyme, and achotinase has an iron sulfur center in its active site. We are going to look at this iron sulfur center in many enzymes in the subsequent reactions. So iron sulfur centers look something like this, right. So I have one slide later on which will show you the different types of iron sulfur centers. So in this case, we have these 3 inorganic ions, and also we have inorganic sulfurs plus cysteine sulfurs which bind these iron sulfur centers. Okay. So once we have this isocitrate formation, it is, then the next step is oxidative decarboxylation, right. So now the second molecule of carbon dioxide goes out. So we have seen the first molecule of carbon dioxide go out when acetyl CoA forms. This is the second step of carbon dioxide that goes out, and this is also an oxidative step, right. So this is oxidative decarboxylation. So the electrons are taken up by NADH. So we have another molecule of NADH formation. Magnesium ion is the cofactor for isocitrate dehydrogenase in this step. So in this case, we have magnesium ion. So from isocitrate, we have alpha ketoglutarate. We are here, right. So now alpha ketoglutarate is converted to succinyl coenzyme A. So again coenzyme A comes in, right. One molecule of carbon dioxide goes out, electrons are taken up by NADH. So coenzyme A, NAD plus, TPP, lipoate, FAD, these are the cofactors for alpha ketoglutarate dehydrogenase. So if you recall from a few slides back, these were the exact same cofactors for pyruvate dehydrogenase. So it turns out that alpha ketoglutarate dehydrogenase and pyruvate dehydrogenase, they are very similar enzyme complexes. So in this case again NADH is released. So you see NADH is released and carbon dioxide is also released. So this is the third carbon dioxide that is formed. So all the 3 carbons of pyruvic acid or pyruvate have been converted to carbon dioxide, right. So its oxidation is complete and the electrons have been taken up by NADH, right. Now this reaction is almost identical to the pyruvate dehydrogenase reaction that we saw a few slides back. It has 3 enzymes which are homologous to E1, E2 and E3 of the PDH complex, right. So since they are very similar, it is hypothesized that most likely these both enzyme complexes have a common evolutionary ancestor. So they must have come from a common evolutionary ancestor. Okay. So we are at this point, succinyl coenzyme A. Now succinyl coenzyme A is converted to succinate or succinic acid by this enzyme succinyl CoA synthetase. Succinyl CoA synthetase is also called succinic thiokinase, right. And this step drives the synthesis of ATP or GTP from, GTP or ATP from GDP or ADP. So GDP is converted to GTP or ADP is converted to ATP, right. So we have basically synthesis of another ATP molecule at this step. Succinate dehydrogenase is then converted to fumarate by the enzyme succinate dehydrogenase. So succinic acid is converted to fumarate by the enzyme succinate dehydrogenase. Now this succinate dehydrogenase is an enzyme which is tightly bound to the inner mitochondrial membrane. We are going to see this enzyme again when I talk about oxidative phosphorylation, right. So, we will come back to this enzyme in more details in the next part of this lecture. So, from succinate fumarate is formed, and again this is an oxidation step. So, the electrons are accepted by FAD, which is reduced to FADH2. Fumarate, this enzyme also has 3 iron sulfur centers, right. So, we have already seen one example of iron sulfur center. This enzyme has 3 such iron sulfur centers. Okay. Fumarate is converted to malate by this enzyme fumarase. It is also called fumarate hydratase. Okay. Now, you will see that there is a double bond here. So, a water molecule is added to form malate. Now, this enzyme is very stereospecific. It will only take fumarate where the double bond is such that it is a trans isomer, right. The cis isomer will not be accepted by this enzyme, and it forms only L-malate. It will not form the D-malate, right. So, this is a very stereospecific enzyme, and it produces only one of the 2 possible isomers. The last step is catalyzed by malate dehydrogenase. So, again these are dehydrogenation reaction, right. So, malate dehydrogenase is formed. It produces, the electrons are again accepted by NADH. So, 3 molecules of NADH are produced, 1 molecule of FADH2 is produced, and this oxaloacetate that is formed in this step will be taken up by the next cycle of this citric acid cycle, right. So, it will again go into this cycle. So, ultimately what happens? The 3 carbons of pyruvic acid are converted to carbon dioxide, 3 carbon dioxide. So, its oxidation is completed. All the electrons are stored up in NADH and FADH2, and also 1 molecule of ATP is synthesized. So, this is a summary of citric acid cycle, where you will see that all these products that are formed. So, these are the oxidation steps. These are the 4 oxidation steps, and in this step as ATP is synthesized, right, and this oxaloacetate which is formed at the last step will again go back into the cycle to start the next round of the citric acid cycle. This table summarizes all the high energy molecules that are produced, starting from glucose all the way to oxaloacetate, right. So, you can see that how many ATP. So, in glycolysis 2 ATP molecules were consumed first. So, we have -1 here, and then NADH is produced, 2 molecules of ATP are produced, and then again we will see, we have seen all the formation of these NADH and FADH2 molecules, right. Now, this NADH will be converted ultimately to ATP in the next part of this oxidation, which is the oxidative phosphorylation, and we will see that depending on the pathway, 1 molecule of NAD can give rise to 1.5 molecules of ATP or 2.5 molecules of ATP. So, 2 NADH will give you 3 or 5 ATP molecules, right. So, if you do this calculation, you will get that from 1 glucose molecule, we get 30 or 32 molecules of ATP. So, 30 to 32 molecules of ATP are synthesized from the oxidation of glucose. Okay. So, what we have seen so far is glucose oxidation is complete into carbon dioxide. However, the NADH still has to be converted to ATP, and the electrons have to be taken up by oxygen to form water, because oxygen is the ultimate electron acceptor in this reaction. So, citric acid cycle, not only it produces or feeds into the final oxidative phosphorylation, but it also produces all these intermediates which are taken up by different biosynthetic pathways. For example, citrate goes into the synthesis of fatty acids and steroids. Alpha ketoglutarate, one of the intermediate molecules, it is converted to glutamate or glutamic acid. This is one of the amino acids, and which is converted to arginine, proline and glutamine. So, all these amino acids are synthesized. On the other hand, glutamate is used to produce purines. So, this will go into the formation of DNA and RNA. Succinyl coenzyme A, it is used to produce porphyrins and heme. So, we have seen this heme, right. It was also present in hemoglobin. Oxaloacetate, it goes into the formation of aspartate, asparagine or pyrimidines. So, purine is here, pyrimidine is here, right. So, 2 more amino acids and also one of the building blocks of DNA and RNA. We have already seen this phosphoenol pyruvate formation, from which glucose is synthesized on one side, and on the other side these other remaining amino acids are synthesized, right. So, all these very important molecules are synthesized from these different molecules that are generated in the citric acid cycle. So, citric acid cycle, just like glycolysis, is also highly regulated, and the basic theme that you will see here is that whenever these high energy molecules like ATP, acetyl coenzyme A, NADH or fatty acid, these are present, they will inhibit these steps. So, this step, this step, this step, this step, all these steps are inhibited by these high energy molecules. On the other hand, when these molecules are present in abundance, so AMP or coenzyme A or NAD plus and calcium ions, then it will activate these enzyme complexes. So, it will activate these reaction steps, right. So, these are mostly allosteric regulation, and the important factor that is needed here is the ratio of ATP and AMP or NADH and NAD plus or acetyl coenzyme A and coenzyme A. So, the ratio of these molecules determines whether a pyruvic acid will go into this citric acid cycle or not. Okay. So, now we are going into the third stage or the last stage of this cellular respiration. So, what we have so far is the form that pyruvic acid, glucose is converted into 2-pyruvic acid, and these pyruvic acid carbons are oxidized to carbon dioxide. So, that step is complete. The electrons have been taken up by NAD plus and FAD, resulting in the formation of NADH and FADH2. So, these are the reduced electron carriers. Now, these electrons have to be ultimately given up to oxygen to form water, and the energy released will be used to synthesize ATP from ADP. So, this is phosphorylation and this is oxidation. So, these two steps are coupled together, resulting in the step called oxidative phosphorylation. So, electron transfer and oxidative phosphorylation, that is what we are going to look at. Okay. So, as I mentioned earlier that most of these things are happening inside mitochondria, right. So, this is one of the organelles in eukaryotic cells. So, let us look at the structure of mitochondria. Mitochondria, they have two membranes, very similar to gram-negative bacteria, right. So, you can see there is an outer membrane here and there is an inner membrane here, right. So, we will talk about gram-negative bacteria in more details in week 9 lectures. The outer membrane is permeable to small molecules which are less than 5000 Daltons, right, and ions, which means that small molecules can easily pass through this membrane, and that is channelized by these membrane proteins called porins. We have already seen porins in previous lectures. The inner membrane is more impermeable, right, which means that it will not allow small molecules, ions and even hydrogen ion to pass through, and this is something that is very important, that even hydrogen ions should not pass through this inner membrane. Molecules such as malate and aspartate cross this membrane via specific transporters. So, any molecule that passes through the inner membrane, they are transported via specific transporters which will allow only those molecules. The inner membrane bears components of the respiratory chain and the ATP synthesis. So, we will see this in more details. The mitochondrial matrix, which is the liquid or the soluble solution part that is inside, it is enclosed by the inner membrane and it contains pyruvate dehydrogenase complex and the enzymes of the citric acid cycle, right. So, whatever you have seen in the previous slides, they all occur inside this mitochondria. So, this is just reference back to porins. This is something that I have discussed earlier. So, this is the membrane and you have these porins. So, these are membrane bound proteins and they have very large diameter so that they can allow molecules of certain size to pass through. So, let us look at the components of oxidative phosphorylation. So, again this is mitochondria and what we are looking at are the two membranes. So this is the outer membrane. So you will have porins in this outer membrane, so that big molecules or molecules which are less than 5000 daltons, they can easily pass through, right? This is the inner membrane, and this is something that is very tightly regulated. So not even hydrogen ion can pass through this inner membrane. And this is where the matrix is. So this matrix is also referred to as N side because it will have a net negative charge, and this intermediate membrane space will be called the P side because it will have a net positive charge. And that net positive charge will be because we will see ultimately that hydrogen ions will be pumped out from here to here by these different parts of this complex. Okay. So there are 4 complexes, 1, 2, 3 and 4. Out of this 1, 3 and 4, they pump out oxygen, they pump out hydrogen ions, which is coupled to the oxidation reduction reaction that happens here, right? So complexes 1 and 2 catalyze electron transfer from NADH and succinate to ubiquinone. So this is succinate dehydrogenase, the same enzyme that we saw in citric acid cycle. So it is a membrane bound enzyme, right? So when it oxidizes succinate to fumarate, the electrons are transferred ultimately to NADH, which is finally transferred to ubiquinone. Okay, so ubiquinone will become ubiquinol. Once this ubiquinone or ubiquinol is formed, complex 3 carries electrons from this reduced ubiquinone, which is actually ubiquinol, to cytochrome C. So this will transferred to cytochrome C, right? And finally, cytochrome C, it's a soluble protein which is present in this intermembrane space. It will go and bind to complex 4 and it will transfer the electrons to complex 4, where the electrons will be finally transferred to oxygen. So the electrons will be taken up by oxygen to produce water, right? So the electron transfers are completed from this NADH to oxygen via these 4 different complexes. Okay, so let's look at complex 1, which is this one. So what it does is it transfers electrons from NADH to ubiquinone, right? So complex 1 is also called NADH ubiquinone oxidoreductase or NADH dehydrogenase. It's a big protein complex. It has 42 polypeptide chains, right? And of which 1 FMN and 6 iron sulfur centers are present. So these polypeptide chains, they bind to these different cofactors, right? So there is 1 FMN and there are 6 iron sulfur centers. Complex 1 catalyzes 2 coupled processes. Okay. First one is hydride ion transfer and proton transfer to the matrix, right? So the hydride ion H minus comes out from here and H plus from the matrix. These are transferred to ubiquinone. So this Q becomes QH2, right? So this is one reaction. So the 2 electrons are taken up by the ubiquinone. So this is the reduction. This gets oxidized, ubiquinone gets reduced to ubiquinol. The second step is 4 protons are transferred from the matrix to the intermembrane space, which is shown by this red arrow. So 4 protons are taken up from here and they are transferred to this intermembrane space. So this is the N side and this is the P side. So these 2 reactions are coupled by this complex 1. And we can write the net reaction like this: NADH plus 5 protons plus ubiquinone results in the formation of NAD plus. 4 of these protons are transferred out from the N side to the P side, and 2 protons, so 1 from here and 1 from here, are taken up by ubiquinone to form QH2 or ubiquinol. So this is the detailed structure of ubiquinone, right? So this is ubiquinone. Okay, so you can see 2 keto groups here. When it takes up 1 electron, then it forms this semiquinone radical, right? So if you put an electron here and then you can push the arrows, so this will form O dot and this will become OH, right? Now you can add another electron, so this O dot becomes O minus and it can bind to this H plus to form OH and OH. So now these two keto groups have converted to two alcohol groups. So ubiquinone, which is fully oxidized, becomes ubiquinol, which is fully reduced. So these are the examples of iron sulfur centers, right? So we have already seen something like this. So this is a more complex iron sulfur center, but it can also exist in this more simpler forms. So iron is coordinated by cysteines or inorganic sulfurs, right? The standard reduction potential of these iron sulfur centers vary from minus 0.65 volts to plus 0.45 volts. So it will also depend on the environment that is provided by the enzyme. Okay. So we are done with complex 1. Now we are looking at complex 2. We have already seen this. So complex 2 catalyzes the electron transfer from succinate to ubiquinone, right? Just like this, here the electron transfers were from NADH to ubiquinone. Here it is from succinate to ubiquinone. In this case, there is no proton transfer. So this is the only complex which does not result in any proton transfer. So this is succinate dehydrogenase. We have seen this in the citric acid cycle, right? And as mentioned earlier, it is a membrane bound enzyme complex. So complex 2 is the enzyme succinate dehydrogenase. It is the only membrane bound enzyme in the citric acid cycle. It has 4 protein subunits, A, B, C and D, right? So this succinate dehydrogenase, it has 4 subunits and it uses these 5 prosthetic groups, 1 heme B, 3 iron sulfur centers and 1 FAD. So this will be the structure of succinate dehydrogenase. So there are 4 subunits A, B, C and D, and you can see that these subunits are labeled here. So this is A, B is here, C and D are here, right? So C and D are integral membrane proteins. So you can see that they are embedded in the inner membrane of the mitochondria. They contain heme B and a binding site for ubiquinone. So ubiquinone is bound to these subunits C and D, and they also have heme B. So heme B, this red molecule that you see, that is heme B. Subunits A and B, they extend into the matrix. So this is A and B, and they are more in the matrix, right, than in the membrane, and they contain 3 iron sulfur centers and 1 FAD bound to it, and they also have the binding site for the substrate. So the substrate will bind somewhere here. Okay. So electrons are transferred to ubiquinone at this point. Now complex 3 comes. So ubiquinone has been reduced to ubiquinol. Complex 3 comes. It will take the electrons from ubiquinone and transfer it to cytochrome C, right? So ubiquinol will be converted back to ubiquinone. The oxidized cytochrome C1 becomes reduced cytochrome C1. So cytochrome C1 is a cytochrome which is part of complex 3, and cytochrome C is a soluble protein which will shuttle between these two complexes 3 and 4. Okay. And while this happens, 4 protons are again pumped out from the N side to the P side, right? So 4 protons from this matrix or the N side will be pumped out into this intermembrane space, which is the P side. Complex 3 is a dimer. So it looks like this. You can see that it is a symmetric dimer, right? And each of these monomers, so if we take only, so if I draw a straight line here, and if I just look at the left half side, that is the monomer. So this monomer contains 11 subunits, right? The functional core, which is shown here, the functional core is also shown here. It has 3 subunits: cytochrome B, which is shown in green, which have the hemes BH and BL, right? So you see these hemes BH and BL shown here. So that is cytochrome B. There is an iron sulfur protein which is purple, right? So the iron sulfur cluster is shown here, and cytochrome C1 which is here, which also has a heme which is shown in red. So this is cytochrome C1. Now this cytochrome C1 is bound to this complex, and this cytochrome C is soluble, which will move out. Cytochrome C is a soluble protein. Now these cytochromes, they have these different types of hemes. So this is something that we have already seen before in case of hemoglobin, right? So in case of hemoglobin, this heme is optimized for binding oxygen, but in this case the heme is present to accept electrons. So it will get oxidized and reduced. So it will shuttle between Fe+2 and Fe+3 states. Now let's look at the fourth complex, which is where the cytochrome C, electrons will be taken up from the cytochrome C and they will be passed into oxygen here, resulting in the formation of water, right? So cytochrome C, which is the reduced form, plus protons from this matrix side plus oxygen, this will get oxidized. 4 protons are pushed out and oxygen is reduced to water, right? Now if you do your balance, you will see that ultimately 2 protons are pushed out, okay, from this N side to the P side, because some protons will be taken up by the water molecule. Okay, so this is complex 4, right? So this is the detail of complex 4. It has 3 subunits, 1, 2 and 3, which are critical for this electron transfer, right? The larger green part that is shown here, it is structural protein and it has 10 additional proteins, right? Subunit 1 has 2 heme groups. So this is subunit 1 shown in yellow. It has 2 heme groups. Subunit 2, which is shown in purple, it has 2 copper ions complexed with the thiol group of 2 cysteine residues, right? So there is copper ion. So in this case, a cofactor is copper. The reaction for this is shown here. NAD plus 11 protons plus half oxygen will give you NAD. So this NADH becomes NAD plus. 1 proton from here is taken up, 1 proton from here and 11 protons from here. So they are all taken up by this oxygen to produce water, right? So you can see this is not a balanced reaction, right? Because we have to count for all the number of protons that are transferred from the inner side to the outer side. So for each pair of electrons transferred to the oxygen, 4 protons are pumped out by complex 1, 4 protons are pumped out by complex 3 and 2 protons are pumped out by complex 2, which is shown here. So for each electron that is transferred from here to here, here to here and finally here to oxygen, right? So the electrons are coming from NADH to oxygen, right? So for each pair of electron going from NADH to oxygen, 4 electrons are pumped out by complex 1, 4 electrons are pumped out by complex 2 and 2 electrons are pumped out by complex, this is 3 and complex 4, so total 10. However, for 2 electrons which are transferred from succinate to fumarate, 4 plus 2, 6 protons are pumped out, right? So for 2 electrons transferred from succinate to oxygen, 6 protons are pumped out. For 2 electrons transferred from NADH to oxygen, 10 protons are pumped out. And that is what is written here, okay? And this is something that will be important to calculate the number of ATP molecules that are ultimately synthesized from oxidation of glucose. Okay. So what we have done so far is we have pumped out protons, right, from the N side to the C side. So we have a huge proton gradient, right? The chemiosmotic theory states that this proton gradient and electric field gradient is the one which stores energy. Okay, so this theory was proposed by Peter Michel in 1961. So it states that the transmembrane differences in proton concentration are the reservoir for the energy extracted from biological oxidation reactions. So the energy that is extracted from the oxidation of glucose is now stored as this transmembrane difference in proton concentration, right? And we can calculate that using this equation, okay? Delta G equals to 2.3 RT delta pH plus F times delta psi. Okay. So now if we plug in these values, where delta pH, the difference in pH between, or the proton concentration between these two sides is 0.75, and the delta psi, the difference in electro potential, right, is 0.15 to 0.2 volts. Okay. So if you plug in these numbers, you get 20 kilojoules per mole of proton. So for 1 mole of proton, the delta G is 20 kilojoules, right. Now when NADH, 2 electrons are transferred from NADH to oxygen, 10 protons are pumped out, right. So 10 times 20, that is 200 kilojoules of this energy is stored in the proton gradient. Okay. 220 kilojoules is the energy that is released from oxidation of NADH. So it means that out of 220 kilojoules of energy that is released when NADH is oxidized to NAD plus, 200 kilojoules of that energy is stored in this proton gradient. Okay. Which means that this is a very efficient process, right. So 200 out of 220 kilojoules of energy is actually efficiently stored. Only 20 kilojoules is lost. Okay. So now we have this proton gradient, right. Glucose is oxidized to carbon dioxide, electrons are accepted by oxygen. However, the energy is stored as this proton gradient across the membrane. Now we have to use this stored energy to produce ATP. Okay. So ADP plus inorganic phosphate will form ATP, and this to make this, so this is not a favorable reaction. To drive this reaction, we will use the energy which is stored across the membrane. So there is this enzyme or enzyme complex called the ATP synthase, which will result in ATP synthesis. So it will allow the passing of proton along the gradient, and it will use that energy to synthesize ATP. So the ATP synthase, it looks something like this, right. So there is a transmembrane region, and there is this part which is in the N side, this is in the matrix side, right. So it will allow proton diffusion from this side to this side. So this is along the gradient, and while this happens, these beta subunits, they will synthesize ATP, right. So when proton goes in, it drives the rotation of this motor. So this is a very fantastic molecular machine, right. And this energy is used to convert ADP into ATP. So protons are going in, there is rotation here, that energy is used to synthesize ATP from ADP. Okay. Now based on the structure that is known till date, this machine is grouped into two, F1 and F2, right. So F1 and FO. So FO is the part which is the integral membrane part, and this F1 part is where the ATP synthesis happens, and it is in the matrix of mitochondria. F1 has 9 subunits of 5 different subtypes: alpha 3, beta 3, gamma, delta, and epsilon. So you can see that there are 3 alpha subunits, 1, 2, 3, and 3 beta subunits, right. And then there is gamma, delta, and epsilon, right. So these are the parts of this F1. Each beta subunit has one catalytic site for ATP synthesis. Each subunit proceeds through 3 distinct conformations: beta ATP, beta ADP, and beta MT, right. So you can see that when this rotates, these 3 subunits have 3 different conformations. One conformation binds ATP, one conformation binds ADP, and the other one is MT. And by rotation, they will bind ADP, catalyze the formation of ATP, and then release that ATP, right. So this rotation results in conversion of these 3 conformations and drives the reaction. The FO complex makes up the proton pore and consists of 3 subunits A, B, and C in the ratio of, there is for 1 A, there are 2 B, and there are 10 to 12 C, right. Now this 10 to 12 C depends on different homologs. The conformational changes central to this machine are driven by the passage of protons through the FO portion of ATP synthase. It is estimated that 4 protons must flow in to produce 1 ATP. So, when 4 protons or 4 hydrogen ions flow in, 1 ATP molecule is synthesized, right. So, now we can do some calculation. When 2 electrons are transferred from NADH to produce NAD+, 10 protons flow out, right. 4 protons flow in to produce 1 ATP. So, 10 by 4 is 2.5. So, oxidation of NADH to NAD+ will result in the synthesis of 2.5 ATP molecules. For succinate, when it goes from succinate to fumarate, 6 protons flow out, right. Now, 4 protons flow in to form 1 ATP. So, 6 by 4 is 1.5. So, oxidation of 1 succinate molecule will result in the formation of 1.5 molecules of ATP, right. So, that is where we get these numbers 2.5 ATP and 1.5 ATP. So, again if we go back to this table, you see that from NADH, so all the succinate are converted to NADH, we will get 3 or 5 because it is calculated as 2.5 ATP per NADH and 1.5 ATP per FADH2, which is coming from succinate. Okay. So, oxidation of glucose will result in the formation of 30 to 32 molecules of ATP. So, for this part you can follow any biochemistry book. Primarily Leninger Principle of Biochemistry is a very good book for this particular lecture. Thank you.